RNA is DNASTAR's software for gene expression analysis, and it offers an optional application within it called QSeq, which is for RNA-seq, ChIP-seq, and miRNA analysis of next-gen data. In this video, I'm going to walk you through an RNA-seq workflow with the end result being gene expression analysis and SNP detection. To begin, we need to import our data. So to do that, click on File, Import Experiments, and that brings up the Project Setup Wizard window. Now notice that there are 4 different experiment types here. So we want to make sure and select RNA-seq gene expression analysis so that the default options and parameters are set up appropriately for us. Then click Next. And this is where we add our reads, and we can do this in a couple different ways. I can either click on this Add Data File button and then navigate to the directory where my reads are saved, or I can drag and drop my files from wherever they're saved directly onto this window. And for this workflow, I'm going to be using 6 different human tissue samples, each containing multiple lanes per experiment. So since I have so many folders of data, I'm going to open up my reads directory using Windows Explorer, and then highlight all of my folders containing the RNA-seq reads and drag them onto my window. Now notice that this is Illumina data, but RNA also supports Roche 454 solid and helicose data as well. Now that my reads have been added, I want to group the lanes by tissue sample. So I'm going to select them all and then click on Merge by Directory. And you can see that that's organized all of my lanes by tissue sample. If I wanted to merge everything into one experiment, I could just select them all and then click on Merge Files. But I'm going to leave them as they are now and then click Next to get to the pre-processing step. From here you can choose the normalization method to be used, and I'm going to leave it with the default of RPKM. And then the next step is to select the template sequences against which the RNA-seq reads will be quantified. I have my templates saved locally, but you can also download the latest version of your reference directly from NCBI by using this download button, and either downloading the entire genome by specifying your organism, or by entering a list of accession numbers for the templates you want to download. But since I have my templates saved locally, I'm going to close out of this dialog, click the Add File button, and then I can select them and add them. Notice that after my template sequences were added, some additional information appears in the bottom section of the window. And this is telling me the feature types in the reference that are going to be used for processing my RNA-seq reads. By default, the most common feature types are used, but you can easily change this list just by selecting something else from the list provided, or you can click in this field and type in additional feature types or remove types from the list. And you can see that as I add additional types, the numbers over here to the right are updated in real time. So I'm going to add exon, gene, and you can see that the number of features is increased. And the same goes for removing the feature types as well. If you wanted to map all of the reads to the reference regardless of feature type, you can just use this Use Sequences as Genes option. One other feature to point out from this step of the wizard is the Configure Advanced Options button. Now, most of the settings that you can adjust from this dialog will not typically need to be adjusted. These are parameters that we've optimized based on the type of project and read technology you're using, but they are available in case you need to modify them. However, something that you might want to use more frequently is the exporting options that you can set up here from the General Settings tab. From here you can choose to export a separate file for unassigned reads. You can also choose to export a wiggle file for visualization using the UCSC Genome Browser, or a GenVision file for visualization using DNASTAR's GenVision software. And you can also choose to export a general alignment file, either as a GFF, a BED, SAM, or BAM file. So once you have the options that you want selected here, you can just click on OK, and then click on Next to start processing your data. Once your data has been processed and your reads have been aligned, the first thing that you'll see is the experiment list view, which allows you to manage all of the data in your project. Now notice you can click on any of these experiments here, and you can see statistics over on the right hand side from the alignment, including the number of MER hits, the number of unique reads assigned, the number of rejected reads, etc. Now, from here you could go a lot of different directions, but what I'm going to do is go ahead and import my gene ontology annotations directly from the Go consortium. So to do that, I select Data, Download Annotations, and that opens up the Download Annotations window. And from here, I need to select my organism, so I'm going to select Homo sapiens, and then click the Download button up here. Now once the file is downloaded, then you click the Import button to import the annotations into your project. So now that our annotations have been imported, the next thing I'm going to do is go to the gene table, which is used to show additional information about the genes in our project, such as expression values, fold change, and annotations. And to see that information, we just need to add some columns using these buttons here on the toolbar. So I'm going to start by adding the expression levels for all of the experiments in our project. And as you can see, you can easily remove a column also just by right-clicking on the column header and selecting Remove Column. I'm also going to add a column for the gene ontology annotations that we just imported by clicking on the Annotations button and then selecting Go ID. And notice that you can get more information about any of these Go terms just by hovering over the term, or if you click on the term, you'll be taken directly to that term on the Go Consortium's website. I'm also going to add a column for fold change, using brain as the control and heart as the experiment. And I'm going to rename this column by right-clicking on the column header and choosing Rename Column. I'm going to name it Fold Change Brain versus Heart. And that's just going to make it a little bit easier later to remember what's in this column. Notice that you can also sort all of the data in the gene table just by clicking on the column header that you want to sort by. And you can also search the gene table by using the search box up in the top right-hand corner. So if I type in a term, I'm going to type in TAF9, press enter. You can see that all of the genes that contain that text are automatically brought to the top and selected for me. So from here, a next step would be to narrow down the genes we're looking at by creating some gene sets of interest. And that can be done in different ways. One simple way to start is by using the filtering tool. And to access that, you go to Filter, Filter All, and that brings up the advanced filtering dialog, which allows you to find genes that match multiple criteria that you specify. So for example, I'm going to search for genes that have the text NADH in the Go ID annotation field and have an expression level in brain greater than 5 and an expression level in the heart experiment of greater than 5 also, and where the fold change between brain and heart is 2-fold higher in either experiment. And then I can click search, and that finds 552 genes. So I'm going to save these results as a gene set by clicking on this button. I'll name it NADH, and I like to paste in my criteria, my search criteria, into the notes field so that it makes it easier to remember later. Then I'll click OK, and that takes me automatically to the gene set list view where I can see the gene set that I just created. And notice that when the set is selected, like it is now, I can see information about that set over here in the details pane, such as the number of genes within the set and the notes that I entered. And I can see details about each gene within the set from the bottom section of the window here. So I can create additional gene sets very simply by returning to the filtering tool and modifying the existing criteria. So I'm going to do that, and this time I'm going to add one more criteria, this time for gene ontology, and I'm going to select molecular function, and I'll search for it. And that brings me down to 15 genes. So I'm going to save those as a gene set as well. And I will paste in my search criteria in the notes field. And that takes me back to the gene set list view, and you can see my new gene set is created here. I'm going to do one more. This time I'm going to change the annotation text we're searching for to ATP, and then I'm going to remove the last two criteria. And that brings me about 1,500 genes. So I'm going to save those as a gene set as well. And that takes me back to the gene set list view. So now that I have several gene sets created, I can use these to create additional gene sets if I want. So if I select all of them and then right click, you can see that I can create a set from the union of multiple sets or from the genes that intersect multiple sets. So that's another way to create some additional gene sets. Another way to find gene sets of interest is to group genes by biological process using the gene ontology view. So I'm going to open this view by selecting Data, Show Gene Ontology. And this view is going to show me all of the gene ontology terms in my project as a hierarchical tree. So notice that I can navigate through the tree by expanding and collapsing these nodes. And if I double click on a term, the p-values and z-scores are displayed for that term, and the genes associated with that term are automatically selected for me. The bar graph over here on the right and all of the other data that's displayed in this view continues to be auto-updated as you navigate through the tree and make new selections. These statistical tools up here at the top are also available to further refine your results. And just as within any view, once you find some genes that you're interested in, you can just click on this Remember Genes button, and that will create a new gene set for the genes that you've selected. RNA also offers many visualization tools to further analyze your genes, and I'm just going to touch on these briefly. So for example, if I return to the gene set list view, I'm going to select all of the genes within the NADH gene set by right-clicking and choosing Select Gene Set in all views. And then I'm going to do a clustering, hierarchical, which will do a hierarchical clustering of the genes I just selected and display them in the heat map view. Now I could also do a k-means clustering on the same group of genes, which by default creates a line graph for each cluster and displays all of them in the line graph thumbnails view. Now you can also access the scatter plot by using the Graphs menu. And notice that you can control the data that's plotted here by using these X and Y axis buttons up at the top. The scatter plot also offers these convenient quick links over here on the right hand side. That's an easy way to narrow down your results further. Now the Venn diagram, which is also accessed from the Graphs menu, is also very useful for comparing the genes within multiple groups. Now the default image here is a dartboard displaying all of the gene sets we've created so far, and I can hover over a specific section to see a summary of those genes over on the right hand side. Now to see a true Venn diagram, which doesn't use more than 3 sets of genes, you click on this button up here, and there's our Venn diagram. And if you had additional gene sets, we only have 3 created right now, you can select them or unselect them by checking the boxes here in this section. And again, you can hover over a particular section of the graph to see details shown over on the right. So once you've finished using the available visualizations and creating gene sets of interest, you may wish to export your RNA-seq reads and reassemble them using SeqMan Engine for SNP analysis. So to do that, I'm going to return to the gene set list view and select one of my gene sets, ATP. I'm going to make sure that it's selected in all views, and then choose Data, Export Sequences. Now from here I need to choose a location for the export. I'm just going to choose my desktop, give it a file name, and then click on the Select button to choose the annotations I want to export along with my reference sequence. So in this case I'm going to choose gene and go ID. Then I want to click on Export Matching Reads. Click on the experiment button and make sure all of my experiments are selected, which they are, and then click OK. And now that the directories for my read files have been added, all I need to do is click on Export and the export will begin. Once my reads have been exported, the new assembler, Seqman Engine, launches automatically, and I just need to click on the Assemble button to begin the assembly. Once the assembly is complete, it will be displayed in Seqman Pro, which is part of LaserGene. Each of the genes will be shown as a separate config, as you can see here. You can sort the list of configs by clicking on the column headers to sort either by name, length, or by the number of sequences in the config. And typically, the higher the number of sequences in a config, the higher the expression level for that gene. Now, to view a config at the sequence level, all you need to do is just double-click on it from this list, and that opens it in the alignment view. To analyze SNPs, you would first select the configs you want to look at from the project summary window. So if I wanted to look at all of the configs in the assembly, I would select unlocated configs, or to look at a single config, I would just select it from this list, and then choose SNP report, and that opens up the SNP report window. Now I'm going to move the SNP report down and also open the alignment view for the same config by double-clicking on it, and now I can see both views at the same time. Now notice that by default, the SNP report shows a summary of all of the putative SNPs in the config that I selected. And you can filter these results in several different ways using these filtering options here at the top of the window. For example, I'm going to edit the SNP% filter by changing the minimum value from 0 to 25%. And so now I'm only displaying SNPs that occur in at least 25% of an aligned column. As I scroll through the SNP report, the SNP I have selected in the report is automatically shown in the alignment view at the same time. And I can double click on any SNP in the report to automatically be taken to that column in the alignment view where I can do editing. Note that you can also view the features that were exported from RNA in the alignment view by opening up the reference sequence by clicking on this triangle here, and the features are displayed here along with their translations. I can also view the config at a higher level using the strategy view, which I can get to by selecting config, strategy view, and this is a more graphical representation of the config. You can see that SNPs appear in this view as hash marks. Now if you're interested in seeing the expression level for a particular gene in the assembly, such as the config that we're looking at now, you can open up the gene table in RNA and search for the gene name, which is by default the name of the config. And you'll be taken to that gene in the gene table where you can see we added the expression level columns earlier. So hopefully that gives you enough information as a starting place for performing your own RNA-seq analysis. If you have further questions, please visit our website at DNASTAR.com or contact us at support@DNASTAR.com.